Unveiling the powerhouse of the cell, the synthesis of ATP introduction. In the intricate world of cellular biology, adenosine triphosphate, or ATP, reigns supreme as the primary currency of energy. From powering cellular processes to fueling muscle contractions, ATP plays a pivotal role in sustaining life. But what exactly is ATP, and how is it synthesized within the cell? Join us on a journey through the molecular machinery responsible for ATP synthesis, as we unravel the intricacies of this essential process. Understanding ATP. Before diving into its synthesis, let's first understand what ATP is. ATP is a nucleotide-based molecule composed of three components: adenosine, a ribose sugar, and three phosphate groups. The high-energy phosphate bonds within ATP store potential energy, which can be released when needed to fuel cellular processes. ATP synthesis. The synthesis of ATP occurs through a complex series of biochemical pathways collectively known as cellular respiration. This process involves the breakdown of organic molecules, such as glucose, to generate ATP through a combination of substrate-level phosphorylation and oxidative phosphorylation. Cellular respiration can be divided into three main stages: glycolysis, the citric acid cycle, also known as the Krebs cycle, and oxidative phosphorylation, including the electron transport chain. Glycolysis. Glycolysis is the initial stage of cellular respiration and takes place in the cytoplasm of cells. During glycolysis, glucose, a 6-carbon sugar, is broken down into two molecules of pyruvate, a 3-carbon compound. This process generates a small amount of ATP directly through substrate-level phosphorylation, where phosphate groups are transferred from donor molecules to ADP to form ATP. Citric acid cycle. Pyruvate produced during glycolysis is transported into the mitochondria, where it undergoes further oxidation in the citric acid cycle. During this cycle, acetyl-CoA, derived from pyruvate, enters a series of reactions that produce carbon dioxide, NAD, and FADH2, electron carriers, as well as ATP through substrate-level phosphorylation. Oxidative phosphorylation. The majority of ATP synthesis occurs during oxidative phosphorylation, which takes place in the inner mitochondrial membrane. In this process, electrons carried by NAD and FADH2 from glycolysis and the citric acid cycle are transferred along the electron transport chain, a series of protein complexes embedded in the membrane. As electrons move through the chain, energy is released and used to pump protons, H+, from the mitochondrial matrix into the intermembrane space, creating an electrochemical gradient. The flow of protons back into the mitochondrial matrix through ATP synthase drives the synthesis of ATP from ADP and inorganic phosphate, PI, a process known as chemiosmosis. ATP synthase acts as a molecular turbine, harnessing the energy of the proton gradient to catalyze the formation of ATP from ADP and PI. Regulation of ATP synthesis. The synthesis of ATP is tightly regulated to ensure energy production meets the demands of the cell. Various factors, including substrate availability, enzyme activity, and cellular energy needs, influence the rate of ATP synthesis. Additionally, feedback mechanisms, such as allosteric regulation and hormonal control, help maintain ATP levels within the cell. Significance of ATP synthesis. ATP synthesis is essential for cellular function and survival. ATP serves as the primary energy source for a wide range of cellular processes, including muscle contraction, nerve impulse transmission, active transport, biosynthesis, and metabolism. Without ATP synthesis, cells would be unable to perform the work necessary for life, leading to cellular dysfunction and eventual death. Conclusion. The synthesis of ATP is a fundamental process that sustains life at the cellular level. Through the interconnected pathways of cellular respiration, cells efficiently generate ATP, the molecular powerhouse that fuels cellular processes and drives biological activity. Understanding the synthesis of ATP provides insight into the intricate mechanisms that govern cellular energy production and underscores the importance of ATP in maintaining cellular homeostasis and vitality.